And welcome you all to the PACK-NIN webinar series. My name is Sudai Sekimuthu. I'm a Marie Curie researcher in Renew Center of Excellence, Slovenia. So this webinar series is conducted through PACK-NIN project. So today our speaker is Dr. Ievgen Pylypchuk. He's a researcher in Department of Materials and Environmental Chemistry, Stockholm University, Sweden. And I introduce about his background, research background. He's from Ukraine, and he graduated from. He did his master's in polymer chemistry, and he did his Ph.D. in physics and chemistry of surface in 2013 at the Chuiko Institute of Surface Chemistry, Ukraine. Then he moved to Poland, and where he did his postdoc and the topic of catalysis and surface chemistry. Then from 2017 to 19, he did his another postdoc in Sweden, in the Department of Swedish University of Agricultural Science. Now he's working with Professor Mika Sipponen group and working on lignin-based nanoparticles and molecular weight characterization. So today the webinar topic is structural and molecular weight dependency in the formation of lignin nanoparticle. So I'm glad to meet every one of you. Thank you. Just one announcement, please. Once you join, and mute your microphone. Thank you. And if you have any questions during the webinar, please post it in chat session. Thank you. Yeah. So please, you can start your presentation. Thank you.
Thank you. Thank you, Suta, for such a nice presentation. And it's a big pleasure to present on your seminar. So please let me know if everything is okay. You can see me and you can hear, right?
Yeah.
And I also see some people I know. So in the end, I will also tell about the NMR protocol for characterization of the surface of nanoparticles we developed recently with our colleagues. So what is the problem? So you know that lignin is very nice material, but the problem that, for example, kraft lignin is highly heterogeneous material. And if you try to prepare nanoparticles from lignin, they will be rather polydispersed. Of course, it depends on like choice of solvent and your nanoparticle preparation method, but in general, they are polydispersed due to heterogeneity of kraft lignin. For example, you see some nice paper. I think it was published in 2018. The particles are nice, but you see also that the size and shape distributions are rather broad. So you can see like small particle, big one, like different shape and size. And if you need to, like, you know, to go into some application, probably it would be better if you have some kind of monodispersed particle, not only by size, but also by shape. And you see the topic of lignin nanoparticles is growing very, very like fastly during especially last years. So when we realized this problem that nanoparticles are highly heterogeneous, we decided like we have tried to solve this question, and we came up with this approach, which was reported previously in, for example, in works of Lavoko, professor from KTH. They use this solvent fractionation technique to fractionate lignin to obtain different lignin fractions, and that's how they reduce heterogeneity. So, for example, here in this sequential solvent fractionation approach, they fractionate lignin with 5 solvents. So they started with ethyl acetate, then followed by ethanol, methanol, and acetone. And on each stage, you can have like this insoluble residue, which you then fractionate with another solvent. And then you have 5 fractions. You see the yields probably by weight is written here. And you have fractions with different molecular weights. So you can see, for example, on this figure that when you go with this ethyl acetate, you have lowest molecular weight, then you go to higher molecular weight with ethanol, followed by methanol, and then acetone. So basically what's happening, you fractionate your lignin by molecular weight. So you have low molecular weight fraction on the first step and high molecular weight fractions on the 5th step. And also you have final residue, which is like contain all it could contain like carbohydrates and also some inorganics. Usually it's not soluble in acetone, poorly soluble in THF, but it is soluble at least in DMSO. What else I wanted to say here? That we have also high aromatic density for low molecular weight fraction, and later on you will understand why it's important. So when we have all our fractions fractionated, we got some results. So for example, here you can see this is eucalyptus lignin, this is spruce lignin. So for example, let's take a case of eucalyptus lignin. You see, when you fractionate from lowest molecular weight to highest molecular weight, you have a lower amount of phenolic units in the end. So you see, it's like gradual number of phenolic units decreasing. So you have highest amount of phenolics in the beginning when you have low molecular weight lignin, and the high molecular weight lignin you have very few phenolic units. And the trend is opposite for aliphatic units. So for example, you see you have very few aliphatic OH in low molecular weight fraction, but when you go to high molecular weight fraction, it's like highly aliphatic or aliphatics rich. And this trend reflects in particle size when you prepare nanoparticles from different fractions of lignin. For example, when you have lower molecular weight fraction separated by ethyl acetate, you have particle size around 200 nanometers. And then when you increase molecular weight of lignin, the particle size is increasing, which is like slightly counterintuitive. Because usually people think that if you have high molecular weight, you should have large particle size, but it's, as you can see, opposite. And actually the same trend we got for spruce lignin fractions. So again, when you decrease, for example, phenolic units, you're getting a lower size of particles, and and it's like the main finding here. So we started to think why it's different. Like since we selected two different lignins from softwood and hardwood, we started to try to make some conclusions why it's different and how lignin structure affect the particle size and other properties. So here you see our method. So it's pretty simple solvent exchange method. When you have lignin solution in organic solvent, then you add water, then you evaporate solvent, and basically you have your nanoparticles here. So what we done, we investigated different initial concentrations of lignin in this solution and their influence on final particle size. So here you see the case of fractionated spruce kraft lignin. So here you can see, for example, this is low molecular weight fraction in this row, fractionated with ethyl acetate, and then we took this fraction and we prepared particles at different starting concentration. And you see, when you increase concentration of lignin from 0.1 milligram per milliliter to 6, you have rather high large particle size in the end. So when you have higher concentration of lignin, of course, you're getting larger particles. So you see trend is similar for all the fractions. But what is interesting that when you have high molecular weight lignin, you have smaller particles. So actually you can see it also on this graph. When you increase concentration, you increase particle size. And when you, maybe it's not very clear here, but anyway, when you increase molecular weight, the particle size is like going down. It's getting lower. So, for example, here we can also maybe comment something on polydispersity of particles. And for example, let's take this fractionated with ethanol. So what you can see is that when you start with very low concentration of lignin, you have small particles, and also you have, I would say, moderate polydispersity. And then if you go, like, this image you can easily see. the particles are rather polydispersed. You see, like the shape is not well defined, and also they're different in sizes. And this is happening when you have like low concentration of lignin in the beginning, probably because of low efficient concentration, so particle cannot like interact properly. But when you increase concentration, for example, to it should be 2 milligram per milliliter, you reach PDI optimum. You see, like you have very low polydispersity, and maybe I can show image of these particles. The image not the best, but anyway, you can see that particles are like more or less defined, and also they are like pretty much the same in terms of their size, or much closer to size to each other by size, and this also reflects in their polydispersity. So what I wanted to say here is that when you prepare lignin nanoparticles, you should take in account also initial concentration, because you can have some polydispersity optimum when you have a proper concentration. Yeah, here I'm showing particles prepared at 4 milligram per milliliter. You see here we have like pretty broad this polydispersity distribution. You see we have small and big particles. And when you go to 6, polydispersity is also high. But what is interesting, you see we have like 2 distinct populations of nanoparticles. One is with the size around 500 nanometers, and another one with size 100 nanometers. So of course, like general polydispersity is higher, but actually you have just mixture of 2 populations of monodispersed particles, which is like also a rather interesting result. So now we can go to Eucalyptus lignin. So the trends are the same. So you see when you increase when you increase initial lignin concentration, you have larger particles. When you increase molecular weight, you have smaller particles. But what is interesting is shape of these particles. You see, so when you have this fractionated eucalyptus lignin, you can get this, I call it nano-donut-like structures. And you can find them almost everywhere, except maybe fraction number 3. I think it's methanol. You don't find that much of these holes inside. Yeah, and also you can see that shape of the size of these donuts increasing when you increase lignin concentration. Yeah, maybe I can tell more on next slide. So again, if you talk about polydispersity of these particles, you see we have some optimum in the concentration range between 2 and 4 milligram per milliliter. For example, if you look on this, like particles extracted with ethyl acetate, we have very interesting result here, because you see, again, when we have very low initial concentration of lignin, we can see that these donuts are not completely formed. You see this like half closed, we can observe here half closed rings, some other shapes, and these donuts not very well defined. You can see maybe here on this enlarged picture. And also we can observe that polydispersity is rather high for this at this concentration. So again, probably since we don't have very much lignin, it cannot efficiently interact one with each other and to form particles. So you should go to higher concentration to get better particles. For example, you see when we go to this 2 milligram per milliliter, where is this polydispersity optimum, we can obtain very nice particles which have this hole through, and like I would say at least 60% of them have this hole inside, and they are rather monodispersed, as you can see here. The same actually result you can get when you go to 4 milligram per milliliter, but when you go to 6, you see you have high polydispersity, and also shape is not very nice already. It's like I would call it, they have like some kind of wrinkled morphology when you prepare them at higher concentrations. Yeah, so the main conclusion I would make to make here, so that the lignin particle size is affected by initial lignin concentration, the shape is affected by source of lignin, and also polydispersity pretty much depend on concentration of lignin. Here you can see this mixed data for both spruce and eucalyptus. And what we can say is that when you have smaller particles, or you intend to get smaller particles, you will have lower polydispersity. And when you go to higher particles, you will have higher dispersity, which is probably obvious. But anyway, interesting result. So usually when I present, people asking me what is the mechanism of this nanodonuts formation. So the clear answer, I don't know. The easiest answer, I don't know. But at least we can have some guesses why it's so. So you see, here we have at 4 milligram per milliliter, I would say that we have this critical micellar formation region. And what I want to say is that probably we have lignin molecules which could look like these micelles, and when they assemble, they can form this kind of nanodonuts. So here we have better image. So imagine they form this kind of cones, and then these cones assemble into this so-called donut structure. And of course, the shape of this like molecules will pretty much depend on molecular weight and lignin structure. And maybe that's why we have these donuts only for eucalyptus lignin, but not for other lignin. Probably like some structure features of eucalyptus lignin cause this kind of shape of molecules, which then pack into this interesting size, shape. But anyway, when we prepared these particles, we knew that we have different structures in lignin. For example, you can see them here. And you see, we can also have some polar structures. And like according to simple logic, probably we should have more polar groups on the surface. And then, if it's true, these polar groups should interact with solvent. And then probably we should get some signal in an NMR experiment. That was our preliminary idea when we started this. So when we first ran NMR of this nanoparticle sample, we got the spectrum. You see, it was almost nothing except water. So water signal was pretty strong, and we didn't know what to do. But then I talked to Per, who is my colleague from KTH, and he developed pretty cool method for water suppression in this NMR experiment. And then actually we got this spectra for lignin nanoparticles. So here you can easily see that we have different units. For example, like here, if you're familiar with NMR of lignin, usually we have this methoxy group of lignin. Here we have some aliphatics group, and here we have some aromatic units. You can also refer to these formulas. Yes, so when we developed this method, we decided to run this experiment for all the fractions we had. For example, here you have spectra of 4 eucalyptus lignin fractions because they were most interesting to us. And when you see on the spectra, they look pretty much the same. But when you try to refer intensity of signals one to each other, you can see that, for example, for this low molecular weight fraction, we have pretty strong aliphatics peak. And also, if you compare this data with TM, you see, for example, again, these particles with most exposed to the solvent holes, holes which are most exposed to solvent were inherent to this fraction number 1, which is here. And you can see also that we have pretty strong aliphatics peak. So then we decided probably this strong aliphatic peak can be correlated with these holes inside. And then we decided that most probably we have aliphatic units inside of the lignin nanodonuts. So you see, we have more of these units inside, more holes, higher intensity in aliphatic region. And these aromatic units, you can see them here. So this was proposed structure for lignin nanoparticles. Actually, we published in SUSMATCHEM and also went to the cover. We were pretty much proud of it with our colleagues. And actually, that's it. So I just want to summarize that lignin nanoparticle size correlate with number of phenolic and aliphatic units in initial lignin. So high aromatic density favors formation of larger nanoparticles. So for example, when you have low molecular weight fractions, but with high aromatic density, you will favor formation of really big nanoparticles. Aliphatic content oppositely lowers aromatic density, and that's why it's also leading to smaller nanoparticles. Particle shape depend on lignin concentration and, of course, to lignin origin. And that's it. I just want to acknowledge my former colleagues from KTH, where I did this work, also my current group in Stockholm University, and also, of course, Sudha, who invited me to give this talk. Thank you. Thank you very much, and ready to listen for your questions.
Thank you, Ievgen, for a very nice presentation. So from audience, if anybody has any question, you can ask Ievgen.
You can. It was a very interesting lecture. Hello.
Yeah, I can hear. Thank you.
Yeah. My question is regarding the donut formation and the source. Like, why is it so particular to the eucalyptus source rather than the other sources? It's quite fascinating and interesting. Could you please give some insights into that? Because what do you feel like? What is different in eucalyptus lignin, and when you compare with the other sources?
Thank you. It's a really cool question, and I still don't have clear answer, but I think at least we have approached it to some understanding why it's so. Probably it's in the structure of like guaiacyl and syringyl units. So when we have, for example, for spruce lignin, we have more of G units, and for eucalyptus lignin, we have more of S units, and probably, like you see, we have this the difference only in additional methoxy group in initial structure of lignin. So this one was for hardwood. So when we go to the shape, because what is happening when we form particles, lignin molecules just somehow pack in the solution, right? They don't like water. They want to stay together. They form these hydrogen bonds between each other. And like, for example, here you see this like model lignin molecule. And probably because we have more of these methoxy groups, they somehow affect self-assembly of lignin in solution. And most probably they have this like, not spherical, but elongated shape. It was also proved by another researchers that lignin in solution is like more elongated or ellipsoidal than spherical. And most probably like this shape of lignin influence packing of lignin molecules. So in my theory, it's like just theory. I don't have any proof of the, I couldn't prove this now, but probably this like, these truncated cones assemble into these donuts. Hope I have answered your question.
Yes, Ievgen, it's interesting. I just have one more curious question. Like, my question is about if we can grow this, self-assemble this around cellulose nanofibers, for instance, or cellulose nanocrystals. Like, will this assembly happen when we have in the presence of cellulose nanocrystals or fibers?
Actually, it's very interesting question. I was also thinking about this, but I never tried.
Okay, because I guess you could develop some beautiful nanostructures, with this kind of growth.
Agree, and I think it's true. Like, it's worth to try. Okay, yeah, maybe one day we should try.
Great presentation, Ievgen. Thanks.
Thank you, Avinash. Yeah.
Any more question? Okay, then I can ask Ievgen. I'm curious about ethyl acetate fractionation. When you explained ethyl acetate fractionation, you said you extracted low molecular weight lignins. So in that case, I believe that because of the polarity difference of ethyl acetate, you couldn't make a fractionation of high molecular weight macromolecular lignin. So in that I believe that in that residue, there was some amount of high molecular weight lignin macromolecule. Did you notice that, or how did you explain that?
I didn't get last point. What should we explain?
When you do the ethyl acetate extraction, you mentioned that you extracted low molecular weight lignin. No?
Yes, all the data were analyzed by SEC,
Okay, but did you like because I did a little work on this ethyl state extraction. So when I did it, I found that because of the polarity difference, I couldn't extract high molecular weight lignins into the ethyl state layer. So that means there are some macromolecules, lignin macromolecules in the residue part. Have you?
So you say that when you were extracting lignin by ethyl acetate, you got some high molecular weight fraction in ethyl acetate layer, but
Not
maybe
in ethyl state layer, in the residue.
Yeah, of course,
Yeah.
because when you extract lignin with ethyl acetate, you get like, for example, on this slide, you extract lignin with ethyl acetate, and then you have residue where you have like high molecular weight fractions, like fractions with higher molecular weight.
Then when you do the ethanol extraction, did you create any kind of bonding with ethanol? Because when you do the ethanol fractionation, there might be some residue ethanol in the lignin layer. Have you ever noticed that?
Yeah, of course. When we do this fractionation, we always freeze dry our samples. And so, for example, when you do this fractionation with ethyl acetate, what you do, you extract it with ethyl acetate, you collect this like soluble fraction, right? And you somehow recover it. Then you have residue. Then you extract this, like you can freeze dry it, for example, and then you extract it with ethanol. And again, you recover this fraction, and this residue you use for extracting this methanol.
Okay.
So it's like, that's why it's called sequential solvent fractionation. So on each stage, you dry your sample, you use another solvent to.
Okay, I understand.
And also you need to dry it because, for example, if you run phosphorus NMR and you have, for example, some ethanol left, it will end up in your phosphorus NMR spectra. So you will have like wrong estimation of aliphatic and phenolic group. So usually it's needed. But again, it depends on your application, why you need to do this. If it's like not crucial, maybe you don't need to dry it completely.
Okay. Okay, thank you for the nice presentation. Is there any question more? If not, I can thank Ievgen. Thank you so much for a nice presentation and sharing the knowledge with us. So thank you all for your participation. See you again in the next webinar. See you. Thank you.
Thank you for the opportunity to present.